Late last month, researchers from the University at Buffalo threw a sort of a monkey wrench into the conventional wisdom that Streptococcus pneumoniae and Streptococcus pyogenes, two common organisms that can cause human disease, survive for short periods of time on fomites. Joining me on the phone is the lead author of the study published in ASM's Infection and Immunity, Dr. Anders Hakansson. Dr. Hakansson is an assistant professor of microbiology and immunology with the UB School of Medicine and Biomedical Sciences. Good morning, Dr. Hakansson, and thanks for taking the time to talk to me from cold and snowy Buffalo.
Well, exactly. Good morning. It is cold and snowy for sure.
I have no doubt. It's about 50 degrees down here in Tampa, Florida, by the way.
Oh, thank you for letting me know, yes.
Yeah, rub it in, right? Okay, well, in the study titled Biofilm Formation Enhances Fomite Survival of S. pneumoniae and S. Pyogenes, you say that the strep species can survive on objects, furniture, toys, etc., much longer than previously thought, thanks to biofilm formation. For my listeners and readers on the website, can you explain what is a biofilm, and are these bacterium always found in biofilm in the environment?
Well, that's a good question. So a biofilm is essentially a bacterial community that what bacteria has been shown to do over many, many years, starting probably about 20, 30 years ago, people have realized that bacteria could kind of aggregate together, and they make this kind of complex architecture with matrix around themselves, and they try to surround themselves to protect themselves against the environment. And inside of this biofilm, in some biofilms, bacteria can have different functions. So some bacteria can do certain things, and other bacteria do other things, so they kind of help each other out. So that's kind of what a biofilm is. And it's only lately, I think the first paper on strep pneumo biofilms came out in 2006 or so, showing that they could aggregate as well. And it's only lately that we've really started to get a good handle on how they make biofilms and where they make biofilms. And what we have kind of contributed to is that we've shown that these organisms actually primarily grow as biofilms when they colonize. And what I mean with colonize is that when they are carried in the back of our nose, they organize that way so that they can protect themselves from us as hosts and live kind of a quiet life there, which is kind of what they probably want to do most of all.
Okay, so but on things like toys in the daycare center, are they always found as biofilms there?
So that's the thing that we don't quite know yet. So our study is one of the first ones to kind of look at it. So the reason we did the study was that in the past, people have known that if you take bacteria that are grown in laboratory media and you put that on a surface, they will die within an hour or a few hours max. Sure. And based on that, people have not seen that as a big threat. So when it comes to pneumonia patients, for example, we treat them in hospitals in a way that once a patient is healthy, we can just wheel them out and wheel another patient in. And we don't have to worry about surfaces because the common sense from the CDC and so on, and the common notion is that these bacteria will die so quickly that we don't have to worry about them. It's a completely different story if you talk about Clostridia or other enteric viruses and stuff like that. Those are known to live on surfaces already, and if a patient is wheeled out of when he's healthy, the whole room will be bleached down and cleaned very, very thoroughly so that they will avoid this surface kind of spread that they know is going on. So the thing is that for a long time, people have thought that these organisms, they won't survive on surfaces. But as we now know that probably the way they survive inside of us is as biofilms, we're assuming that when we sneeze and cough, what is coming out is probably also biofilm bacteria. And as biofilm bacteria are known generally to be much harder and much more resistant to environmental stress, we're curious whether those would survive better on surfaces than the experiments that have been done in the past. And that's kind of essential what the study finds.
Right, and your study took place in a daycare center, if I'm correct.
Yeah, so we actually started the study in the lab, just kind of trying to see if we take the bacteria from laboratory media, put them on plastic and dry them down, or if we take the biofilm bacteria that we can make in the lab and put those down on plastic, how long will they survive? And we realized that the bacteria from laboratory media, they die in hours. And the biofilm ones, we realized they actually survive up to at least a month, which was much longer than we actually thought. And then next we figured that another way of spread that is common is hands, of course, that we get things on our hands and then we can spread that onto an object or onto someone else's hands, and that could be another spread. And hands have a tendency, our skin produces a lot of molecules that kills bacteria. So bacteria don't survive for a long time on skin. And if we put then the laboratory media grown bacteria, they will be dead in a few minutes. But we can actually, the biofilm bacteria can be surviving for several hours on hands. And then we really became interested in the daycare setting, that we know that these bacteria are found either in schools, they're found, of course, in hospitals, but a daycare setting would be kind of a very good way to look at these, because that's where they spread around a lot. So we went into the daycare looking for surfaces that are hard to clean, like soft toys or books and other types of surfaces. And what we found was that we could indeed culture these bacteria quite readily on all of these surfaces.
Okay, and you're saying that Streptococcus pneumoniae may survive for a month on these surfaces.
Exactly.
So, of course, this changes everything. I mean, these bacterium can cause disease in humans, and sometimes very serious disease. So the longer the organism is viable on the surface of an object, the more chance for person-to-person spread and infection, particularly for the young and the old.
That's true. So the only thing I want to say is that in one sense it's not kind of that this study will make things people more sick. This is something that has been probably, if it is going on, it has been going on for a long period of time. So it's not really changing anything. The only thing it changes is that if the spread, we don't know exactly how much spread is occurring through surfaces. So a lot of spread is probably occurring through droplet spread when we sneeze and so on, the way that people, the notions that we think of when we think about these bacteria. But there is a possibility that a fair amount of spread actually does occur through surfaces. And if we understand how much that is, and if we get more vigilant with cleaning our hands primarily, I would say, before we eat or before we interact with objects and so on, we might actually be able to reduce the spread that is currently going on and reduce then infections. So,
right.
So that's the way we look at it.
Yeah. Do you have any plans for some follow-up research on this topic?
Well, personally, I'm thinking about exactly how to do this. So I'm not an expert in doing epidemiological studies, but I think that if this information now is available for people that do studies about spread between individuals, if they could incorporate, looking at surface spread as one possibility, we might learn a little bit more about how common this type of spread is. And I think that might actually inform us about if we should implement kind of the recommendations that are used for other bacteria that do actually are known to spread through these types of surfaces and so on. And if we should do that, then, if the spread is high, then we should definitely do that. And we should be more vigilant in daycare settings and in school settings about cleaning surfaces and so on. And I think hand washing is one thing that we can already do now, and I think that would be helpful not only for these bacteria, but for a number of other viruses and bacteria that spread around in schools and daycares.
Yeah, this is clearly a fascinating study and definitely opens up a lot of questions. I'm going to go ahead and that'll be the end of it. I'm going to close with that. I want to thank Dr. Anders Hakansson with the University of Buffalo. I appreciate it, sir. Great research.
Thank you so much. It was nice to be talking to you.
All right. You take care now. Bye-bye.
Bye-bye.